Today we're going to talk about advanced genetics. Advanced genetics is any type of inheritance pattern that does not follow your typical Mendelian inheritance. So as mentioned, not all traits follow a strict Mendelian pattern of inheritance. That's when you're thinking there's a dominant and there's a recessive, and the dominant will overshadow the recessive trait always. There's other possible patterns, and we're going to talk about all of the ones listed: codominance, incomplete dominance, multiple alleles, sex-linked, and polygenic traits. To start with, let's get into codominance. This is a type of inheritance pattern where neither allele is dominant, so both will show up at the same time if it's a heterozygote. Let's talk about an example here. Purple and white flowers. When the genotype is homozygous, right, two P's, the flower is going to be purple. If it's homozygous with the W's, then it's going to be white. But if it's heterozygous, you're going to get purple and white flowers, which look like stripes. So let's look at our first Punnett square here. We're crossing a homozygous purple flower with a homozygous white flower, and our offspring are all PWs. That means they're all heterozygous, so our genotype ratio is 100% PW. And our phenotypic ratio is 100% purple and white flowered. Just looking at the offspring, that's what we get. Now, if instead of crossing two homozygous purple and white, we cross two heterozygotes, we're going to get a much more mixed up batch of offspring. So now, looking at just the offspring here, you can see that we have some homozygous purple, homozygous white, and heterozygous. So our genotypic ratios here, 25% PP, 50% PW, and 25% WW. And that's strictly looking at just the four offspring boxes. For our phenotypic ratio, we're just going to determine what those genotypes stand for. So 25% are purple flowers, 50% purple and white, and 25% just white. So if codominance is when you see both of the alleles, incomplete dominance is when neither allele is dominant. And so when the two alleles combine to make a heterozygote, you actually make a brand new phenotype. An example of this are snapdragon flowers. You have red flowers, you have white flowers, but when red and white come together, they actually make pink flowers. So a brand new, never before seen phenotype. Looking at our Punnett squares here, again, in our first one, we are crossing a homozygous red flower with a homozygous white flower. Looking at just the four offspring, you can see that they are all the same. So in this case, we have 100% R prime, or what that would show is 100% of a pink flower. Now, if we were to cross two heterozygotes, or two pink flowers, our genotypic and phenotypic ratios would look a little bit different. Our genotypic ratio would show that we have 25% of our offspring showing RR, 50% showing RR prime, and 25% R prime, R prime. Again, only looking at the four offspring here. Our phenotypic ratio is just looking at what the genotypic ratio is and determining what that actually stands for. In this case, 25% red flowers, 50% are pink flowers, and 25% of the offspring will be white flowered. Two additional types of non-Mendelian inheritance are multiple alleles and polygenic inheritance. Multiple alleles is just the idea that two or more alleles are possible for a gene. And what this means is that you could have multiple alleles that code for the same gene. An example of this is blood types. Blood types have three alleles, IA, IB, and lowercase I, and with those three alleles, you can make four different blood types. And those are listed in the bullet points below. But if we stop for a second and look at this Punnett square, you'll notice that we have our one parent being IA lowercase I, which is type A blood, and our second parent being IB lowercase I, which is type B blood. They are heterozygotes, right, because they don't have the same alleles for both of their alleles, so they are heterozygotes. And when we look at the offspring, you can see straight off the bat that they are all different. So our genotypic ratio, looking at these 4 offspring, is 25% IAIB, or type AB blood, 25% IAI, like one of the parents, 25% IBI, like the other parent, and 25% lowercase I, lowercase I. When you're looking at these genotypes, again, you're just taking those genotypes, looking back at this sort of code that we have created, and our phenotypic ratio is showing that it's 25% of each of the blood types, 25% AB, 25% A, 25% B, and 25% O blood. Polygenic inheritance is when you have multiple genes controlling a trait. So this is when you have genes that have 2 alleles each, but 2 or 3 or 4 different genes controlling a singular trait. This allows for a ton of different phenotypes. So think about eye color and skin color and height. There's not just 1 or 2 phenotypes there, right? There's tons of different eye colors, tons of different skin colors, and this is because those traits are controlled by many genes. And last but not least is our sex-linked traits. Sex-linked traits are traits carried on the X chromosome, so only the X chromosome. Now, something special about this is the difference between a biological female's genotype and a biological male's genotype. Since a biological female has 2 X chromosomes, they can be considered carriers. That means that they can carry, right? They can have a defective allele and a functional allele and not exhibit whatever the trait is, right? They can kind of push that aside as if it's a recessive trait. But they can also exhibit the phenotype. They can show whatever the trait is if they have 2 defective X chromosomes, 2 defective alleles on the X chromosome. Now, men are XY, right? So they only have 1 copy of the X chromosome. So they cannot be carriers because if they have a defective allele, they have to show it because they don't have a second X to hide away that defective allele. So biological females can be carriers because they have 2 copies of the X chromosome, but biological males can only either have the trait or not have the trait because they only have 1 copy of the X chromosome. Some examples here are like color blindness or going bald or hemophilia, which is a blood typing disorder, a blood clotting disorder. Now again, looking at these different genotypes, you can see that XX is just a normal biological female, not colorblind. If you have the prime here, that means you would have the trait. In this case, you're a carrier because you have 1, but not both. Same on the third one, except now you have color blindness because both of the X's show the defective allele. And then when we get into biological males, XY, that would be normal, and then the X prime shows that you are colorblind. So looking at this Punnett square down below, we are crossing a carrier and a dad who has color blindness. So the carrier is XX prime. Again, a carrier can only be female, and then a dad who has color blindness. Looking at the offspring here, you can see that there are 4 different genotypes that are present. You have 25% are a female carrier, 25% are female colorblind, or X prime, 25% XY, 25% X prime Y, and the phenotypic ratio, you would have to specify whether it is female or male. In this case, 25% carrier female, 25% colorblind female, 25% normal male, and 25% colorblind male. So again, I'm getting all of this just from analyzing the Punnett square, and we're going to do a little bit more of that in class. So great job.